The overall goal of this procedure is to perform rapid and efficient targeted genome editing in the AAVS1 of human pluripotent stem cells, or human PSCs, using a flipase recombinase-mediated cassette exchange approach, or RMCE.
RMCE improves the targeted insertion of a sequence into the AAVS1 locus, which is of great importance for stem cell biology as it allows rapid testing of the function of a molecule by overexpression or knockdown.
The main advantage of this technique is that human PSC lines, free of random integration events, still pluripotent, and with normal karyotypes can be created in just 15 days.
This method provides a useful tool for transgenesis in the AAVS1 locus, but can also be applied to other human permissive loci.
To begin, culture human PSC master cell lines under standard procedures on or off inactivated mouse embryonic fibroblasts, or IMEFs, in 6-well plates. When the cells reach 60 to 70% confluency, plate drug-resistant IMEFs by first using 0.1% gelatin solution to coat the necessary wells of a 12-well plate and incubate the plate at room temperature for 5 minutes. Then plate 125,000 IDR4s per well and incubate the cultures overnight with IMEF medium. The following day, pre-incubate human PSC cultures with fresh medium, including 10 micromolar ROCK inhibitor at 37 degrees Celsius for 1 hour.